Yeah, good evening, everyone. I hope my audio and visuals are clear as of now. Yeah. Um, Anyone who is watching the live session can just text me on my WhatsApp that the audio, visuals, everything is fine because here I cannot get like if it's okay or not. So please, if anyone is watching it, please let me know in my on my WhatsApp that everything is fine or not. So yeah, today I will be discussing the membrane transport again. That is the third part where I will be focusing on certain active transport and ion channels. So we are going to discuss the membrane transport. We are going to discuss the membrane transport, and that is part three today. And today I will finish the membrane transport in general physiology. I will finish the membrane transport in general physiology, and we are focusing on the cell membrane transport, and we are now going to discuss active transport and one of the types of active transport, that is vesicular transport. We were discussing active transport yesterday. We were discussing active transport yesterday. And in active transport, now we will focus our major discussion on vesicular transport. Vesicular transport. Now, when I say vesicle or vesicular, it means there are certain vesicles. If there are certain vesicles, okay. So remember that vesicular transport basically it is a type of active transport. So it is a type of active transport. It is a type of active transport. Apart from that, you have to focus on the part that it requires calcium for its functioning. It requires calcium for its functioning. And apart from that, it requires some special proteins, or it requires some special or docking proteins. Requires special proteins. Requires special proteins. So we have focused our discussion on, we have started our discussion on vesicular transport. That is again a type of active transport which I didn't discuss yesterday. So it is a type of active transport, and this vesicular transport basically requires calcium and some special proteins. Some special proteins. Now remember guys that the vesicular transport, it is basically of two types. It is basically of two types. It is exocytosis and we have endocytosis. Endocytosis. So we will be discussing each of these terms in detail. In detail, we are going to focus on each of these terms in detail, guys. So I hope the audio and visuals are perfectly fine, guys, and you guys are no able to see. You guys are able to see, and yeah, anyone can text me like on WhatsApp. Whosoever is watching it live, just give me a thumbs up in my WhatsApp like number, so that I'll get an idea that everything is fine at my end. Everything is fine at my end. Yes. So now if we focus our discussion on exocytosis. 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 You guys are very well aware with the fact that suppose this is a cell. This is a cell. And in this cell, you know, there are various vesicles which are present. Various vesicles which are present. Yeah, there are various vesicles which are present, and these vesicles वेसाइकल्स का काम होता है स्टोर करना, कुछ सब्सटेंसेस को स्टोर करना, और मोस्टली न्यूरो ट्रांसमीटर्स होते हैं. So yes, mostly they are the neurotransmitters which are stored in the vesicles, तो इसके अंदर कुछ कुछ न्यूरो प्रेजेंट होंगे। तो ऑब्वियस सी बात है, ये आप लोग का जो रिलीज पार्ट है, कि ये रिलीज कैसे होगा, तो यू कैन जस्ट फोकस ऑन द पार्ट कि ये जो सेल है, ठीक है, दिस वेसिकल विल मूव टुवर्ड्स द सेल मेम्ब्रेन। आपको पता है ये वाला पार्ट क्या होता है, सेल मेम्ब्रेन। दिस इज कॉल्ड एज द सेल मेम्ब्रेन। So this vesicle will move towards the cell membrane. Okay, this vesicle will try to move towards the cell membrane so that it can release into the synapse, right? So okay, drawn it with black color. So I'll draw it with black color. Yeah. So yes. Now when it will move towards the cell membrane, guys, this vesicle will now fuse with the plasma membrane of the cell. Okay, and it will get burst, and these chemicals will get released into the synapse. These substances will get released into the synapse. Okay, so you can every now if you just see, the normal cell membrane is there, and vesicular membrane is also coming and joining this cell membrane. This is the cell membrane, this is the cell membrane, and this is, what is this, this is the vesicular membrane. Vesicular membrane. Now you can very well see that vesicular membrane is combining with the cell membrane. So in turn, what I can say that it is increasing the total area of the cell membrane. It is increasing the total area of the cell membrane. It is increasing the total area of the cell membrane. It is increasing the total area of the cell membrane. So Why? How it is increasing? Because the vesicular membrane जो है वो combine कर जाती है cell membrane के साथ, तो automatically जो area है cell membrane का वो बढ़ जाएगा. So this type, it is this process, it is known as exocytosis. And for this particular mechanism, what all is required that you have to like mention in your notes? For exocytosis, what all is required? For exocytosis, what all is needed? Remember, we require calcium. I have already told you. Secondly, we require ATP. Why ATP? Because exocytosis is a type of active transport, vesicular transport. In turn, it is a type of active transport. तो एक्टिव ट्रांसपोर्ट के लिए एटीपी की जरूरत तो होगी ही होगी, कैल्शियम की भी जरूरत है। और मैंने आपको बताया था कुछ स्पेशल प्रोटीन्स हैं जिनको हम लोग डॉकिंग प्रोटीन्स बोलते हैं, जिनको हम लोग डॉकिंग प्रोटीन्स बोलते हैं, डॉकिंग प्रोटीन्स। So for exocytosis, we need some special proteins or docking proteins. Now, what are those docking proteins or special proteins? You have to mention these in your notes. These is something which is relevantly, like frequently asked in the exams. This is something which is asked frequently in the exams. So remember the very first one that you have to remember, it is syntaxin. Syntaxin, or it is also known as the T snare protein. It is known as the T snare protein. Secondly, you have to remember it is synaptobreviin, or it is also called as the V snare proteins. Now, these are some special proteins which are required for the functioning of these vesicular transport. Okay, V snare proteins, and the third one that you have, to remember, guys, it is one of the important, that is snap protein. It is snap 25. It is snap 25. So all these three examples, like, you have to memorize. It is syntaxin, also called as the T snare protein, synaptobreviin, also called as the V snare protein, and snap 25, and snap 25, snap 25. Now, one important point that I have to like focus in exocytosis on the next slide. Yeah. You already know that this is the end part of the neuron, and this is the neuromuscular junction that I'm going to show you right now. NMJ, everyone knows that this is the neuromuscular junction. Neuromuscular junction. It is the neuron, it is the muscle, and this is the neuromuscular junction. Synapse. Now, there are some toxins. There are some toxins, and these toxins are called as botulinum botulinum toxin. These toxins are called as botulinum toxin. Now these botulinum toxins, what they do? Now these botulinum toxins. Now what they do, beta? They will try to block. They will try to block these docking proteins. They will try to block these docking proteins. Okay. Now what will happen? Everyone knows that in the vesicles inside this neurons, what is there? In the vesicles, there is what is present over here, guys. I've already told you in the neurons there are some substances, these are called as neurotransmitters, and what is there? It is ACH in the vesicles. Now what does ACH stands for? It is the acetylcholine. It is the acetylcholine which is required for, okay, muscle contraction. But yeah, acetylcholine will not release. Now what these botulinum toxins will do, they will try and block these vesicular proteins over here. So obviously there will be no release of acetylcholine or ACH. There will be no release of acetylcholine or ACH. And over here you guys will see flaccid paralysis in the patient. Flaccid paralysis in the patient. Muscular contraction होगा ही नहीं, paralysis हो जाएगी. So this is the flaccid paralysis that you will encounter in these particular cases. So that was one clinical significance that you have to remember about the vesicular transport, that is exocytosis. Exocytosis. Now these exocytosis is basically of two types. Now if we just try to see, this exocytosis is basically, or I can say there are two pathways. There are two pathways of exocytosis. One, it is the non-constitutive or regulated pathway. Non-constitutive or the regulated pathway. And the other one, beta, it is the constitutive pathway. It is the constitutive pathway. Okay. Now, in the non-constitutive or regulated pathway, remember that there is synthesis of hormone or enzymes or neurotransmitters or neurotransmitters, and after they are being processed, now after they are being synthesized, they are being processed, they are being storage, and release further they release. So yes, it is about the नॉन कॉन्स्टिट्यूटिव. इसमें प्रोसेसिंग भी होगी, स्टोरेज भी होगा, और उसके बाद क्या होगा, रिलीज हो जाएंगे. तो सिंथेसिस, प्रोसेसिंग, स्टोरेज, रिलीज. कॉन्स्टिट्यूटिव पाथवे में बिल्कुल उल्टा है. इट इज लाइक जस्ट the steps are missing over here. So you should remember that there is only synthesis and they are directly released. They are directly released. Okay, they are directly released. Now we'll try to tabulate the examples also. So if we just try to see the examples of non-constitutive or regulated pathway, or examples of constitutive pathway. Now, in non-constitutive or regulated pathway, the very first example that you have to remember, it is the secretion of insulin by beta cells of pancreas. Everyone knows that, right? It is the secretion of insulin by the beta cells of pancreas. The second example that you have to remember, it is the secretion of glucagon. These are hormones, insulin, glucagon, pancreatic hormones are there. It is by alpha cells of pancreas. Pancreas ये ना बीटा अल्फा सेल्स पेनक्रियास में ही है. Okay. Now there is secretion of pancreatic enzymes by acinar cells of the pancreas. Acinar cells ये हम लोग डिटेल में पढ़ेंगे जब हम हार्मोनल फिजियोलॉजी पढ़ेंगे. So don't worry about it. We will discuss it in detail when we will discuss the hormonal physiology. And remember, one more example, that is secretion of serotonin by Raphe Magnus nucleus. So it's a kind of a nucleus, okay, and it is involved in the secretion of serotonin. It is involved in the secretion of serotonin. तो ये सब हम लोग जब नर्व फिजियोलॉजी पढ़ेंगे तो उसमें हम लोग और अच्छे से डिस्कस करेंगे. हार्मोनल फिजियोलॉजी में सारे पेनक्रियाटिक एंजाइम सेल्स वगैरह सब बहुत अच्छे से डिस्कस करेंगे. जस्ट नाउ यू हैव टू इन जनरल फिजियोलॉजी में आपको बस अभी एग्जांपल्स पे फोकस करना है. ठीक है, जब टाइम आएगा तो हम लोग इसको बहुत अच्छे से डिस्कस करेंगे इन डिटेल. Apart from that, guys, remember the examples of constitutive pathway, that is secretion of immunoglobulins by plasma cells, IgA, IgE, IgG. तो ये जितने भी इम्यूनोग्लोबिन्स होते हैं वो प्लाज्मा सेल से निकलते हैं और उनका सिक्रीशन, डायरेक्ट सिक्रीशन और रिलीज हो जाता है. कॉन्स्टिट्यूटिव का मतलब क्या है? सिक्रीशन के बाद तुरंत रिलीज. यहाँ पे क्या है कि आप लोग को पहले इसको सिंथेसाइज करना पड़ेगा, प्रोसेस करना पड़ेगा, स्टोर करना पड़ेगा. सिंथेसाइज, फिर प्रोसेस, फिर स्टोरेज, और फिर क्या आ जाता है? रिलीज. तो इसमें लॉन्ग प्रोसीजर है नॉन कॉन्स्टिट्यूटिव में. कॉन्स्टिट्यूटिव क्या? डायरेक्ट है, सिंथेसाइज किया और रिलीज कर दिया. और दूसरा एग्जांपल इसमें आप लोग को यू हैव टू मेंशन दैट देयर इज सिक्रीशन ऑफ कोलेजन बाय फाइब्रोब्लास्ट. Secretion of collagen by fibroblast. Secretion of collagen by fibroblast. So yes, that was about the non-constitutive pathway, or it is also called as the regulated pathway. Whereas we have the constitutive pathway, in which we have focused our discussion on the two major examples. So we have discussed about exocytosis. We have discussed exocytosis in detail. Now I will focus my discussion on endocytosis. Endocytosis. So put the heading as endocytosis. Endocytosis. Now, guys, for endocytosis, for example, this is a cell. This is a cell, and this is a substance which is present outside the cell. This is a substance which is present outside the cell. For example, you have heard the term amoeba, right? Amoeba grab its food through endocytosis. Now, what this cell will do, this cell will move towards this particular substance, okay? This particular substance, this cell will try to grab this substance, okay? This cell will try to grab this substance, yes? And this substance, it will try to engulf it. It will try to engulf this substance, okay? Now, what will happen? Now, what will happen? This will engulf the substance, and this part of the membrane will cut from the original part. Okay, this is the cell membrane, right, guys? This is the cell membrane. Now, cell membrane will try to engulf it, and after engulfing, it will like detach from the plasma membrane. Okay, it will detach from the cell membrane, and it will form a vesicle kind of a thing. It will form a vesicle kind of a thing. So you can very well see what will happen next. It will come inside. So in this case, the total area of the cell membrane is reduced because cell membrane is involved in grabbing this particular substance. As soon as it grabs it, okay, it will detach from the cell membrane. It will detach from the cell membrane. So a part of the cell membrane is lost over here. A part of the cell membrane, it is lost from over here. So it is like decrease in the total area of the cell membrane. Decrease in total area of the cell membrane. So this was endocytosis. In endocytosis, guys, for endocytosis, what all things are required, you have to like mention in your notes. For endocytosis, what all is needed? It is calcium. It is calcium, ATP, clathrin. Clathrin is basically the special proteins. Okay, clathrin is basically a type of a special protein which is required for endocytosis. And remember, it is actually a receptor mediated endocytosis. Endocytosis. Receptor मैंने आप लोग को पहले भी बताया था. I've told you before also, it is present on the surface of the cell membranes. So it is like kind of a receptor mediated endocytosis. Apart from that, remember there is one more protein which is required, that is called as caveolin. ये सारे names आप लोग को याद करने हैं because this is something which is very very important over here. So this is the name of the special protein or the docking proteins which are required. And caveolin, basically it is present in endothelial cells. It is present in endothelial cells. Endothelial cells, and this is also responsible for absorption of nutrients. It is also responsible for absorption of nutrients. It is also responsible for absorption of nutrients. Apart from that, there is one more protein which is like special protein which helps in endocytosis. It is called as dynamin. Dynamin, or it is also called as pinchase. Pinchase. Now, this protein, that is the dynamin or pinchase, it basically helps in helps in attachment of sorry, it's not attachment, it is detachment. It helps in detachment detachment of vesicles from cell membrane. It helps in detachment of vesicles from cell membrane. ठीक है, वेसाइकल्स को सेल मेम्ब्रेन से दूर करने के लिए, ठीक है, डिटैच करने के लिए जिस प्रोटीन की रिक्वायरमेंट होती है दैट इज कॉल्ड एज डायनामिन, क्योंकि जैसे ही ये सेल मेम्ब्रेन से अलग होने की कोशिश कर रहा है तो इट इज फॉर्मिंग अ वेसाइकल, और ये वेसाइकल किससे जुड़ा हुआ है, सेल मेम्ब्रेन से, तो इसको डिटैच करने के लिए जिस प्रोटीन की जरूरत है दैट इज डायनामिन और पिंचेस, दैट इज डायनामिन और पिंचेस. Now, apart from that, guys, remember the next part that we have to like focus our discussion on. We have discussed endocytosis, exocytosis. One more cytosis that you have to discuss, that is like a combo of both, that is called as transcytosis. That is called as transcytosis. Transcytosis. Transcytosis, or it is also called as cytopempsis. It is also called as cytopempsis. Now, what is happening? Example you have to remember over here, that is colostrum. Colostrum, basically it is present in the mother's milk, और उससे ही क्या synthesize होता है, immunoglobulins. The first immunity जो बच्चे को मिलती है that is derived from the colostrum that is present in the mother's milk, and these immunoglobulins basically they enter the cell, they enter the cell, okay, via the process of endocytosis. They enter the cell via the process of endocytosis, and from the cell, these immunoglobulins are released into the blood. They are released into the blood vessel. This is blood vessel, and they are released by the, okay, they are released by the process which is called as exocytosis. Exocytosis. So that's why it is called as transcytosis. It is involving both the process, endocytosis also and exocytosis also. Okay, it is involving both the process endocytosis also and exocytosis also. So yes, so we have endocytosis as well, we have exocytosis also over here. Now, this was about the transcytosis or cytopempsis that you guys have to remember. And yes, so we have discussed the active transport in detail, and yesterday we have discussed the two types of active transport, primary and secondary. Apart from that, guys, I have focused my discussion today on the vesicular transport, which is again a type of active transport, which is of three types: endocytosis, exocytosis, and transcytosis. Transcytosis. So this completes the active transport. Passive transport already I have completed in the first lecture. Now a special topic that I have to discuss a little about over here, that is regarding the ion channels. That is regarding the ion channels, and this is somewhat a part of the transport only. This is somewhat a part of the transport only. Now, ion channels. Now, remember that the movement of ions through ion channels, through ion channels is passive. आप लोग को बताया था मैंने, पैसिव ट्रांसपोर्ट है, और पैसिव में से भी कौन सा है, डिफ्यूजन. इट इज बाय डिफ्यूजन. ओके, दैट इज फ्रॉम higher to lower concentration. That is from higher to lower concentration. Okay, without requiring energy, क्योंकि पैसिव है तो एनर्जी की रिक्वायरमेंट तो नहीं होगी. without requiring energy, or a carrier protein, or a carrier protein. तो ना कैरियर प्रोटीन की जरूरत है, क्योंकि कैरियर प्रोटीन जहां पर यूज होगा वहां पे एनर्जी की जरूरत होगी। तो ना तो यहाँ पे हमें एनर्जी चाहिए विदाउट रिक्वायरिंग एनर्जी और एनी कैरियर प्रोटीन, इट इज from a region of higher concentration to lower concentration, and that too by the channels. These are called as ion channels. It is passive and it is taking place by the process of diffusion. It is taking place by the process of diffusion along the concentration gradient. Okay, तो डाउनहिल ट्रांसपोर्ट भी बोलते हैं इसको, डाउनहिल ट्रांसपोर्ट भी बोलेंगे इसे, राइट। Now we have type of ion channels. We have type of ion channels. I will discuss in the next slide because it is a little bit long. Okay. So put the heading as types of ion channels. Now the very first type that you have to focus over here, it is voltage gated. Now voltage gated, you have to remember it actually open when there is a change in the membrane potential. Membrane potential क्या होता है, we will discuss in detail. So these are the changes in the membrane potential. Okay, and after basically the channel opens, channel openings take place. Apart from that, remember there are something which is called as ligand gated. Okay, ligand एक सब्सटेंस होता है जो सेल मेम्ब्रेन पर आकर बाइंड करता है। तो इसमें जो चैनल्स हैं, which channels which open response to in response to ligand binding, ligand binding। ओके, अब ये ligand एक्सटर्नल भी हो सकता है, बाहर से भी आकर बाइंड कर सकता है, और इंटरनल भी हो सकता है, इंटरनल भी हो सकता है। एक्सटर्नल में एग्जांपल याद रखिएगा, न्यूरोट्रांसमीटर, न्यूरोट्रांसमीटर और हार्मोन्स। हार्मोन्स बाहर से आके बाइंड करते हैं, न्यूरोट्रांसमीटर्स comes from outside and binds, and internal we have एग्जाम्पल्स लिख लेना इसमें, इंट्रासेलुलर कैल्शियम, इंटरनल मतलब अंदर की तरफ होगा, तो इंट्रासेलुलर कैल्शियम आप लोग को याद रखना है। कैंप साइक्लिक एडिनोसिन मोनोफॉस्फेट, लिपिड्स और जी प्रोटीन। ओके, और जी प्रोटीन विच इज प्रोड्यूस्ड in the cells can bind directly to channels and activate them. So there are certain G proteins which can bind to the channels directly. Okay, we have CAMP, lipids, and intracellular calcium, which are internal ligand gated, and external we have neurotransmitters and hormones. Neurotransmitters and hormones basically they act from outside, whereas when we talk about the intracellular calcium, CAMP, lipids, or G protein, they act from inside. G proteins are produced by the cells and they can directly bind to the channels and activate it, right? Apart from that, guys, remember the third one. It is phosphorylation gated. It is phosphorylation gated. Okay, phosphorylation का मतलब होता है फॉस्फेट ऐड करना या फॉस्फेट रिमूव करना। फॉस्फेट ऐड करोगे तो भी चैनल ओपन होगा, रिमूव करोगे तो भी चैनल होगा। तो यहाँ पे जो फॉस्फोराइलेशन होती है वो प्रोटीन की होती है। तो रिमेंबर वेयर प्रोटीन फॉस्फोराइलेशन, फॉस्फोराइलेशन और डीफॉस्फोराइलेशन, dephosphorylation, regulate opening or closing. Where protein phosphorylation, basically प्रोटीन से फॉस्फेट आके ऐड हो जाएगा तो उसे प्रोटीन फॉस्फोराइलेशन बोलेंगे, और प्रोटीन से फॉस्फेट रिमूव हो जाएगा तो उसे प्रोटीन डिफॉस्फोराइलेशन बोलेंगे। तो फॉस्फेट ऐड और रिमूव होने की वजह से प्रोटीन के साथ चैनल ओपन भी हो सकता है और क्लोज भी हो सकता है। तो दैट इज द चैनल्स विच डिपेंड ऑन द फॉस्फेट मॉलिक्यूल्स, the channels which depend on the phosphate molecules. One more type that we are going to discuss, it is stretch pressure graded. The fourth one, it is the stretch pressure gated or mechanosensitive. Mechanosensitive कहने का मतलब है where mechanical stretch mechanical stretch of the membrane results in opening. Results in opening. Where the mechanical stretch of the membrane results in opening. तो ये कुछ आयन चैनल्स के बारे में था जो आप लोगों को याद रखना है। तो आयन चैनल्स वोल्टेज गेटेड मतलब मेम्ब्रेन पोटेंशियल पर डिपेंड करते हैं, लाइगैंड गेटेड एक्सटर्नल इंटरनल, फॉस्फोराइलेशन प्रोटीन फॉस्फोराइलेशन पे डिपेंड करते हैं या डीफॉस्फोराइलेशन पे, और स्ट्रेच जब आप मैकेनिकल फोर्स लगाते हो, ठीक है। तो ऐसी तरीके से मैकेनिकल स्ट्रेच होती है सेल मेम्ब्रेन में, उसकी वजह से चैनल ओपन हो जाते हैं, ठीक है। So that was about the types of ion channels. That was a short discussion on ion channels as well. Apart from that, the last topic for today's lecture, which I want to discuss with you guys, I want to, uh, it is, yes, yes, it is about the ion channels. Okay, specificity. Remember that the ion channels, it can be specific for sodium, potassium, chlorine, and calcium. Okay, it can be specific, it can be non-specific, it can be non-specific. Okay, basically these permit movement of any ions, movement of cations and anions. तो इससे कोई भी पॉजिटिव चार्ज और नेगेटिव चार्ज इजीली जा सकता है। तो कुछ चैनल्स बहुत ही स्पेसिफिक है, कुछ स्पेशल एटम्स के लिए, और कुछ बहुत ही ज्यादा कुछ स्पेशल आयंस के लिए, और कुछ नॉन स्पेसिफिक है through which any cation or anion can move, right? So remember that each channel exists in multiple forms with diverse properties. Now, for example, we have chlorine channels. Chlorine channels जो है, they are actually dimers. They are made up of two subunits, and these are found especially in bacteria, plants, and animals. These are chlorine channels which are actually like they consist of the two subunits. These are called as dimers, and they're found in bacteria, plants, and animals. Apart from that, remember we have sodium channels, including the epithelial sodium channels जो एपिथेलियल सेल्स पर प्रेजेंट है, तो उसको हम लोग ई एन ए सी भी बोलते हैं। काफी बार एग्जाम्स में ई एन ए सी दिया जाता है। दीज आर एपिथेलियल सोडियम चैनल्स, दीज आर एपिथेलियल सोडियम चैनल्स, ओके। बेसिकली दीज आर ट्राइमर्स। इनमें तीन सब यूनिट्स होती हैं और तीन सब यूनिट्स आप लोग को याद रखनी है, दैट इज अल्फा बीटा एंड गामा। अल्फा बीटा एंड गामा। देन वी हैव पोटेशियम चैनल्स, then we have potassium channels. Remember, these are tetramers. These are tetramers. Basically, they are four subunits. फोर सब यूनिट्स होते हैं और बीच में एक सेंट्रल पोर होता है, ओके, फॉर्मिंग अ सेंट्रल पोर, फॉर्मिंग अ सेंट्रल पोर। तो ये बीच में एक सेंट्रल पोर देखने को मिलता है और चार सब यूनिट्स होती हैं। तो वी हैव फोर सब यूनिट्स। देन वी हैव एक्वापोरेंस। Aquaporins. So basically these are also tetramers. These are also tetramers, and these are basically having a pore in each element. Having a pore in each subunit. Okay. तो कहने का मतलब है कि हर सब यूनिट में आपको एक पिट दिखाई देगी, so it is forming a pore in each subunit. Okay. And additionally एक सेंट्रल पोर भी देखने को मिलेगा। तो एक पोर तो हर एक यूनिट में होगा और साथ में एक सेंट्रल पोर भी प्रेजेंट होगा, ठीक है। अपार्ट फ्रॉम दैट वी हैव ligand gated, we have ligand gated, okay, it is such as acetylcholine receptor, okay, GABA A receptor, GABA A receptor, and glycine receptors, glycine receptors. And remember, all these receptors ligand gated, these are pentamers. They are having five subunits, pentamers. The last one, that is chlorine channels. और क्लोरीन चैनल्स अगर हम ह्यूमंस में बात करें, इन ह्यूमंस, उस टाइम पे मैंने आपको पिछली वाली स्लाइड में बैक्टीरिया, प्लांट्स और एनिमल्स बताया था। ह्यूमंस में क्लोरीन चैनल्स एक्सिस्ट एज पेंटामर्स, दे एक्सिस्ट एज पेंटामर्स, दे एक्सिस्ट एज पेंटामर्स। So guys, this was our today's topic discussion of ion channels and the other types of active transport, which I have already discussed with you people. So I have completed, guys. So I will just wind up the lecture. But before that, guys, remember that I have already completed the cell membrane transport, cell membrane transport. So in general physiology, beta, I have already completed the cell membrane transport. And cell membrane transport, all the three parts, part 1, part 2 and part 3, these all are completed in detail now. I will be giving you certain MCQs that you will solve by yourself. I will be sharing the file or the PDF on my Telegram channel. Moreover, if you have attended the today's lecture, please submit your notes at my Gmail, docsman@gmail.com, so that I can know like how many students are really paying out attention and listening to me very carefully. So the first topic in general physiology is done. Tomorrow I will start the second topic, okay? That is cellular fluids. Cellular fluids, okay? So these cellular fluids topic I will discuss tomorrow. So thank you for your patience. Okay, for listening me out, guys. This is something like keep on enjoying, guys. Please start studying at your end. Otherwise, you will lack up in a very different scenario. Thank you so much. God bless.